for joining tonight. Today we will be discussing Zooxanthellae resistance to coral bleaching using HSP70. My name is Jeffrey Connors, and this is my third year in BioBuilders Club. Tonight I am joined by my co-authors.
Hi, I'm Nadia Shah, and I've been doing BioBuilders for 3 years.
Hi, I'm Abaya Gervedi, and this is my second year at BioBuilders Club.
My name is Ava McCone, and this is my third year at BioBuilders.
All right. If that's it, I will hand it over to Ava, and she will start a discussion.
So the problem that we are currently addressing is coral bleaching. And what exactly is coral bleaching? Well, coral bleaching is when ocean temperatures are higher than average for an extended period of time. When this occurs, small organisms that reside in coral, or zooxanthellae, are ejected from the coral. Zooxanthellae is what gives corals their pigment, and when they're discharged, this color goes away, and the coral is therefore bleached. This is significant because these creatures are essential to maintaining the health of the reef by nourishing it with sugar and oxygen produced by photosynthesis. And without these creatures, reefs have become prone to deterioration. Over the past few decades, the effects of climate change have been worsening, and as students and environmentalists, we are very concerned about our planet, and in particular the ocean, as it is home to billions of organisms. As a result, we applied our knowledge of synthetic biology to create resilient zooxanthellae.
So when choosing a chassis for our project, we considered a mutualistic relationship in which multiple organisms work together to ultimately benefit a system. Since the ocean temperature plays a large role in our construct, we decided on creating a heat-stable variant to work with the zooxanthellae that make up the coral, and found that Symbiodinium sp would be the most efficient chassis. Symbiodinium sp are microalgae that are also a type of symbiotic zooxanthellae. They are well known for their mutualistic relationships with coral reefs and can be considered indispensable to preventing coral bleaching. We intend to use a temperature-induced promoter to initiate our construct. Once the ocean temperature is higher than average for a period of 4 weeks or longer, it will take effect. Next, we plan to use the HSP70 gene to regulate the sensitivity of zooxanthellae to temperature. HSP70 is a type of heat shock protein, which are a group of proteins produced when cells are exposed to different stresses, a prominent one being thermal stress. When the temperature of a system increases, HSPs have the potential to regulate the sensitivity of cells and prevent them from reacting to a temperature change. In our case, the HSP70 gene will be expressed in the Symbiodinium sp, and it will provide a heat-stable variant alongside the existing zooxanthellae. We will conclude our construct with the terminator.
Now I'll move to discuss possible experiments that we could use to test the construct. We propose an experiment in which we take 2 samples of zooxanthellae and put them in containers of water that mimic the pH of the ocean and are a bit higher in temperature than their normal habitat, the Great Barrier Reef. We would leave the 2 groups in these conditions for a month, after which we would compare the proportion of coral that underwent coral bleaching in each respective group. We would compare the 2 proportions using a 2-proportion z-test to see if our construct had statistically significant results. If it did, we would conclude that our construct works, and if it did not, we would know that we would need to go back to the drawing board to alter the construct to better obtain these results.
Additionally, the use of our chassis, Symbiodinium sp, will allow for the safe creation of our GMO. In nature, this form of dinoflagellate already has a symbiotic relationship with the coral, and thus, by using it as a vessel as our genetic material, we would not be introducing new organisms into the aquatic ecosystem. Additionally, we chose to use a temperature-induced promoter because when the temperature of the surrounding water returns to its average, the promoter will cease to initiate biological activity. This essentially would prevent an overproduction of HSP70. Coral reefs are endangered worldwide and will only continue unless change is put to action. With our project, we hope to address this critical issue and ultimately preserve ecosystems around the world.